you want to do it.
Okay, I think that we're going to go ahead and get started. So thank you everybody who showed up to our spring seminar series. Today I have the distinct honor of welcoming Dr. Eva Harris, who is the professor and chair of the Division of Infectious Diseases and Vaccinology, as well as the director of the Center for Global Public Health and chair of Infectious Disease and Immunity, PhD program at the School of Public Health at the University of California at Berkeley. She's developed a multidisciplinary approach to study molecular virology, pathogenesis, immunology, epidemiology, and the clinical aspects and control of dengue, Zika, chikungunya, and the most prevalent mosquito-borne viral diseases in humans. Her work addresses immune correlates of protection and pathogenesis and viral host factors that modulate disease severity using in vitro approaches, animal models, and research involving human populations. So thanks, and please welcome Dr. Harris.
Okay, great. Thank you so much. Thank you, Chris. Thank you, Mark. It's really nice to be back here. I think it was like 2018 or so when I was here, and that feels like universes ago. And even pre-pandemic was kind of like behind the Iron Curtain, you know. It's kind of crazy, but it's really nice to be back. And I'm going to be telling you kind of a top date. And I gave a story that initially I thought like, oh my goodness, I talked kind of in this arena. I wonder what I said last time, and it's like totally different. I mean, we really advanced a lot despite COVID. And some of the work I'm presenting is just freshly in press and others from before. So I'm going to be telling you about this continuing saga of immune imprinting of dengue and Zika, and kind of looking at both immunological, epidemiological, and some clinical consequences of this. And it does unfortunately have a lot of implications in terms of vaccine development as well. Anyway, so I'm going to go through this and then hopefully have time for some questions. And so dengue and Zika, well, dengue anyway, we're going to focus on first here as a major public health problem. Half the world population is at risk. It's mosquito-borne by these Aedes aegypti and Albopictus mosquitoes, which are ubiquitous and are domestic, meaning they live and breed around your home in clean standing water, and they're a major problem everywhere. Dengue is the most prevalent mosquito-borne viral disease in humans, and up to 1,500 million cases a year. And then this kind of annoying, terrible problem of what's called dengue hemorrhagic fever and shock syndrome, or severe dengue now, where perfectly healthy people can go into this vascular leakage syndrome and die within 24 to 48 hours. And so what ends up happening is that since we don't really understand, fully the mechanisms and the risk factors, then when you have these giant epidemics, the whole population is petrified and ends up with like major disruptions socially, to the medical system, to families. I mean, it's a huge problem. And so the problem has been also this there's an echoing in the sound. Should be good now. Is it okay now? Okay. Anyway, so this increase in the number of cases, these are decades and these are number of cases. The geographic spread, this is hemorrhagic fever in the Americas prior to 1981 and afterwards. And the same thing is happening in South Asia. It's always been a problem in Southeast Asia, but now South Asia, India, Africa. So it's just everywhere. And then one of the biggest issues is that there are these four flavors, these four serotypes, 1, 2, 3, and 4. And they used to be kind of in different geographic regions, but now, and they're just all four serotypes are everywhere. And unfortunately, the greatest risk factor for severe dengue is prior infection with a different dengue serotype. So what I'm going to be talking to you about is unpacking that at the immunological and virological level. And so the clinical manifestations are, we call it like Ebola, or breakbone fever. And actually, out of curiosity, I can't see the hands on Zoom, but how many people have had dengue? Okay, do you remember it? Yes, you do not forget it. And you're the lucky ones who survived, because I think that's why you invited me, right? Anyway, so but it's like acute onset febrile illness, really, really painful bone and muscle ache. But the problem is, again, with this kind of movement of some few, the unlucky few, into what we call hemorrhagic fever and shock syndrome, where the hemorrhagic is true, and there are petechia, and then later going to have gross bleeding manifestations. But the real issue is this increased vascular permeability. And then also, okay, and just to mention that in 2009, I think you were part of this effort, but there was a reclassification by the WHO, and it became called dengue with and without warning signs and severe dengue, which was a way to kind of aid in triage and really in clinical management. But we still use both definitions of severity because they have kind of different parameters, and some are more important for case management and others for more pathophysiology studies, you know, so as you see when I walk through this, we'll be keeping both of those going. And so in terms of the genus, so flaviviruses are right here, the 4 flavors of dengue 1, 2, 3, 4, closely related to Zika, and this antigenic, phylogenetic and antigenic similarity will become clear with the problem is, but also yellow fever, and then the more neurological, Spesnile, St. Louis encephalitis, Japanese encephalitis, et cetera, and then the tick-borne encephalitis as well. And so it's a 40 nanometer particle, and this is what we're seeing it as a mature particle, which actually, there's several states. And so here, when it's a mature virion, we have this envelope protein, which forms these antiparallel dimers, all the way, there's 180 monomers, 190 of these dimers that are completely across the whole viral surface. When it gets into host cells, into the host endosome, with a pH lowering as it goes to the late endosome, the reorientation of a conformational change of these antiparallel dimers into these essentially trimeric, what look like spikes, which expose the fusion loop, which is at the tip here in green, and which will allow it to fuse with the endosomal membrane and then spill its contents out. And then you get essentially cytoplasmic, really in these vesicle packets, translation of this positive strand mRNA, which is the viral genome. And so it's essentially mRNA kind of ready to rock and roll, and then it just gets translated and it makes more of itself, which is packaged, and then it's assembled with the structural proteins, envelope PRM, and then the capsid with the RNA, and then the non-structural proteins, including polymerase and helicase, and really importantly, this one secreted protein called NS1, which we have shown, in fact, Rentia Chang, who's here in the audience, the brilliant undergrad who's now here and as a graduate student, she was really part of the team that was showing how that NS1 protein causes vascular leak all by itself. And so there's a way that you can have this immunological triggering essentially through, I'll tell you in a moment, but essentially through antibody-dependent enhancement, and you can also have this direct viral pathogenesis. So there's an immunopathogenesis and there's a viral pathogenesis as well. Okay. And so there's all this stuff going on, right? So you've got individual risk factors, what I call epidemiological risk factors, that have to do with the number of susceptibles in your population, and the hyperendemicity, like what are the serotypes that have been in your population before? Because if you've had dengue1, you will generally be protected from disease with dengue1, which is susceptible to all the other 3, but you can't tell that easily because of the cross-reactivity, you know, at the serological level. I will show you how some really cool stuff if we get to it about kind of unveiling what's going on in the inapparent infections. Then you have viral risk factors at the serotype level, but things like Vaccinology. I mean, the serotypes are genotypes, and the genotypes are clades, and inside the clades are strains. And we've shown that the severity can actually differ based on the clade. And so there's a lot going on here. And then we've got NS1 thrown into the mix. So it's a very complex disease. And then we have Zika, which, as you know, really exploded in the Americas. Initially was at the island of the Gap in 2007, went to Polynesia, and then entered. And when it entered this entire continent, which was essentially had seen Zika, so it was 100% susceptible with this Aedes aegypti mosquito, which is what transmits Zika. It just went like wildfire, and within just 1 year, it completely spread through the entire continent, causing this really frightening microcephaly and congenital Zika syndrome. Otherwise, it's quite mild. I was really worried when it started coming in because it was gonna mess up our serology, which it did. But no one saw coming. Actually, other flaviviruses, there are some sheep flaviviruses which have been associated with abortions and such in sheep, like miscarriages and whatnot. But no one ever was thinking about this in relation to a flavy. It's like a really big problem. And so my essentially, my PhD is actually niche genetics of all things, and so I come from a very molecular background. And so when I started working on dengue as a huge public health problem, the other side of my life has been devoted to capacity building, and so I was kind of both doing this basic work, and then I took a break and started a huge whole capacity building from the bottom up in Latin America, and then I wanted to connect the two. And when I connected the two, I kind of came back to the lab, but of course what I knew was molecular and cellular, and so I kind of started there, knowing that I had to work on dengue because it was such a big public health problem, but having like zero epidemiology, virology, or immunology, or clinical, or everything. So it's been like a deep degree learning curve my whole life. And so I started kind of inside the cell with molecular virology, but then, this is an immunopathogenesis, so you need an animal model, you need, but animal, of course, mouse is not the usual host for dengue, so you need to work in human populations. So, kind of now it's really this nice system where we can go back and forth. And what I'm going to tell you about is kind of the work based on our cohorts in Nicaragua, and what we've learned about immune correlates and about this imprinting at an immunological level, one of the clinical and epidemiological immunological consequences. All of this is not possible, it wouldn't be possible without this huge collaboration. I've been working very closely for 35 years. I just came back 2 days ago. I go every other month, and it's a wonderful system and incredible collaborators. And we have the National Virology Laboratory and a nonprofit that we founded, which has it's headquartered here and in Nicaragua. And we have a hospital-based study, and then this what really important work on is this cohort. When Zika came along, we worked in pregnant populations, and we do a lot of work in communities as well. Including interventions that went through a whole cluster randomized control trial, et cetera. So kind of really the spread, kind of integrated disease holistic view of the infectious disease. So now finally, we're going to the presentation now. It's not over yet, it's just starting. All right. So we're going to do an introduction to the cohort, and I'm really going to talk about imprinting. About everything around antibodies. And so what I'm going to show you first is the effect of prior dengue infection on dengue, and then a prior Zika infection on dengue, kind of dengue writ large. Then because of the epidemiology as our cohort unfolded, we were able to start breaking that down to look at the effect of prior dengue, but by incoming serotype. And then Zika on dengue by incoming serotype. Okay, so that's kind of like as we're kind of unveiling this story. But now, this is really cool. We can now address the hidden landscape of interparent infections with this new multiplex serological tool we've developed. And so now that allows us to look back and suddenly start understanding all of the interparent infections by serotype. So it's kind of like the next frontier is looking backwards, but I'm going to show you the differences in circulating serotypes and symptomatic infections, and then differences in clinical outcome by serotype. And what I want you to get from this is how friggin' complex it is. And really, that these are not dengue. These are 4 different viruses. They really have different characteristics, and it's really important to think about it that way when we think about how those 4 viruses interact with the 5th, which is Zika, and then with others as they come in. And we can talk about that later. So just the cohort study is, I think it's now in its 20th year, and it's 2004 where we founded it, and we initiated 4,000 children ages 2 to 17, and it's in Managua, which is the capital. And we added chikungunya in 2014 and Zika in 2015. And right now there's an active about 4,000 children, but to date there have been around 9,400 participants. And we analyze them annually, as you'll see. So we have like 70,000 titrations of antibodies that we've done across all these years. And the children stay in for generally around 5 to 10 years. So we can really look at this very cool, the repeat infections, in this one population. And so what happens is that we take these annual samples, and then we follow the healthy samples, and then we follow anything that looks like a dengue, chick, or Zika by the case definition of the WHO. We also follow undifferentiated febrile illnesses that are not anything else, and then we work all of that up using PCR and molecular and serological tools and virus and virus isolation to identify that symptomatic disease. But because we have these annual samples, we also can capture inapparent infections by looking at the titer of antibodies side by side every year, year 1, year 2, year 2, year 3, and if there's a 5-4-fold increase in titers or a seroconversion, then we know that they've had an infection. And then if they come in, then we have a case. If they don't come in, then that was an inapparent infection. And then the cool thing is that we can go back in time because we have 1.2 million samples now, and there's literally 50 chest freezers in Nicaragua, and there are 50 different, tanks for liquid nitrogen and the PBMCs, and we can go back in time and look at, the pre-infection sample about someone who later developed an inapparent versus symptomatic or a severe versus for non-severe disease. And that idea of kind of new correlates or biomarkers of both protection and risk is what we've been able to do over these years. And so this allows us to look at antibody repertoire dynamics, at at comorbidities, at repeat infections, homotypic infections at the B cell level, and then in the pre-infection sample, we can look at immune correlates of both protection and pathogenesis. And we look at mostly antibodies, as I mentioned, in B cells, and there's functionalities like neutralization, and you often think of neutralizing antibodies, but there's also a lot going on the FC portion of the antibody. We call that FC effector functions, in which we found also have a really important role in both protection and pathogenesis. And so what I'm going to talk, this is kind of a lot of where our work is. What I'm going to talk about here is really this imprinting in relation to pathogenesis, and then kind of how we identify these repeat infections. And so this is just looking at the cases of dengue that we've had in Nicaragua, and you can see over the years from 24 to 2023 here, that we have generally, one dominant serotype per year. And so this is like dengue 2, and then dengue 3, and then dengue 1. And then we had this giant double epidemic of chikungunya when it came in. Chick is an alpha virus, but it's also transmitted by the same mosquito. Is Egypti, and then right after that we had this giant and very intense Zika epidemic, and then kind of the stunned silence across the Americas. This was literally 2.5 months, and it infected 60% of the population. And then we had a huge dengue 2 epidemic, which allowed some really interesting epidemiological and immunological studies. And then COVID, and then this really cool kind of suddenly when the borders opened, we got 3 new viruses coming in, and in fact 3 new lineages, because by then we could do our own sequencing and phylogenetic analysis in Nicaragua, which was the gift, if you want to say so, of COVID, was to get sequencing into all of our sites. And we did this through a partnership with our colleagues in Ecuador, who were able to transfer the technology virtually during the pandemic for SARS, and then we moved that into dengue. So it was a really nice story. So now let's start looking at what, how these all match up. So the idea is that this concept of immunopathogenesis. So what happens is you have, say, one infection with dengue 1, and then you get a primary infection, which is generally inapparent asymptomatic or mild disease. Mild is mild, right? I mean, dengue is really intense, but we call it mild because you're not dead. And so then you get, the B cell, T cell response, all this good stuff, memory B cells, memory T cells. And so when you get the same thing again, in fact, you can get homotypic infections, and we've actually found a ton in our cohort. It's just that people don't look at them usually because they can't capture them. But they generally don't have a huge burden, but you can get them, but you don't get really sick from them. So so that gets cleared. But when you have another serotype that comes in, you actually can get this enhanced infection. And why is that? So it's called ADE. And what is that? So generally, dengue, this little green guy over here, will infect myeloid cells, which are essentially like monocytes, macrophages, and dendritic cells. And so you'll get a certain amount of infection. So after that infection, you'll have green antibodies to the green virus. Okay, so you'll have like anti-dengue 1 antibodies to dengue 1, but they actually will cross-react with the next serotype coming in because it looks kind of similar, right? You saw that phylogenetic tree. And so then you'll get an immune complex but with a one-on antibody. And so what that can do is that not only can the flu virus go into the cell with this cognate receptor, but the immune complex can go into the FC receptor, which will bind to the FC portion of the antibody that's complex to this new virus. But it's not actually neutralized. So the virus is fine, and it's just like, yo, take me alive, and it goes in. So you end up having this opsonized infection. So it's actually more infection. And so, and there's epidemiological, underpinnings for this, and the way we measure this in the lab, and is that we, can take a cell which has no cognate receptor, and it only has an FC receptor, and so will only be infected in this scenario. And then we add an antibody, to dengue or an isotype control where nothing happens. And what happens is that when you have zero antibody, you don't get infection. When you have a perfect amount of antibody, you get this peak of infection, which is allowing in through the FC receptor. When you have a lot of antibody, eventually you will block that. And this is called the ADE curve. And so a lot of forces to happen with many viruses, but this actually happens in vivo, and that's what we were able to show. When Leah in the lab, who now has her own lab at NIH, was able to show that, okay, so we're looking at antibody titers bin, fourfold titrations in the x-axis, and the hazard ratio for dengue hemorrhagic fever and shock syndrome. And what you can see is that at a certain, this is compared to naive, right? So you can see that at a 1 to 21 to 80 evolution of, and this is in children, right? This is not in vivo. So children who have those titrations that we do painstakingly every year, 4,000 pairs, and this is the titer that they have. So at a certain titer, you have this peak, actually this like 7-fold risk of having DHFDSS, which you don't have at lower or higher amounts of antibody, okay? And so, and you actually, can see this also with this other definition. This is the most like narrow definition of severe disease, and so you kind of get this higher peak. You can see it for hospitalization, but you don't see it when you look at all the dengue cases or just any dengue case, which is really interesting. Now, when you look at that with a continuous hazard ratio, then you see again, this looks very similar to that ADE curve that I showed you in vitro, but this is actually the children's own antibody levels against their future risk of dengue hemorrhagic fever and shock syndrome, okay? And interestingly, the same relationship was seen, exactly the same titer in a Thai study that was published right after, in the year after ours. And so this is like, it actually happens in real life. And so that's dengue on dengue. So now that Zika came in, we were interested, so everyone was worried, like what's the relation between dengue and Zika? And so after we had Zika in 2016, we kind of waited around as you do in epidemiology, you kind of wait for the next thing to happen, and then bam, we got this giant dengue 2 epidemic, and that allowed us to look at the relation of Zika and then the dengue 2. And so this is a little complicated, but what we're able to do is because of following these children year by year by year. So if you just take someone here and you look at their antibody levels, it's a pretty mess. Like you cannot tell what they had before or anything. It's just a big bunch of cross-reactive antibodies of dengue and Zika. But the beauty of this cohort study is that we have every year, and so we can capture, oh, this was a primary dengue 1, and so we know what their dengue history is. And mostly like if they've had or not had dengue or Zika. So we can create naive, the dengue, 1 dengue, 2 dengues, 2 plus dengues, 1 Zika, 2. So we can create this immune history for each child. And then what we can look at is the probability of symptomatic dengue. And so you have 1 dengue. We know that dengue, I just told you, is a risk for second infection, which we can see here, especially the dengue 2. This is the dengue 2 epidemic in 2019. 2 dengues, however, is not a risk. So if you had 2 already, you're actually, if not protected, you're neutral, okay? Interestingly though, if you had 1 Zika, you're at risk of symptomatic dengue. And if you had dengue and then Zika, you're still at risk. Okay, so here it goes down, but here it's still at risk. And that's just for symptomatic dengue. And so now what about severity? So when you do severity, you can see that you have, again, Zika is a risk factor for both styles of severe disease, okay? And it was interesting because if you remember that, like in that plot that I just showed you, well, you'll see in a moment. So this allows us, if we put all these together, and the reason dengue 4 is black over here is we never had dengue 4 for like, 19 years as a cohort. So we were looking at dengue 1, 2, 3, and Zika, and wondering about dengue 4. What I showed you right now is that Zika is this increased risk for dengue 2. So that's the one thing we know so far. Okay, so now let's start looking at serotypes. So in that same paper, which is the one I think you guys read for this, right? Maybe I don't need to say anything because you all know. No, I'm just kidding. Okay, so now we're gonna grade it by serotype. So this is what we did not do in the previous paper, right? And what you see here is this ADE curve of dengue. When you had a dengue incoming, only dengue 2, we have that same ADE curve, whether you have symptomatic dengue or severe dengue. And what's really interesting is that what I showed you before was that there was not a risk for just dengue case when you put all those serotypes together. But now that you start pulling them apart, you see that there's a risk for dengue 2, symptomatic dengue, that you didn't see when it was a big mish of all the others. So what about, this is like after Zika, but now we're saying, what about, let's just look at what happened when we had dengue circulating. Because before 2016, it was only dengue. So we're only looking at dengue now, and we see the same thing. A little wonky, but we see here that we get symptomatic dengue. We have that ADE curve again with these binned antibody titers 21 to 80 is where we see this risk phenomenon. Okay, what about the other serotypes? So now if you go to the next thing, whoops, that was supposed to be. So the crazy thing, if you look at dengue 1, it's completely different. Okay, so this is the curve of, this is like 0, this is naive. And you can see that when you have a dengue 1 incoming infection, it actually is protective at all antibody levels. Okay, so it's really different. Like these are not the same virus. And we can talk about why I think that's going on at the biological level. But this is, and then when you look at dengue 3, you get this funny thing where for a symptomatic dengue, it's actually protective at high titers, but you actually get this curve of enhancement for severe disease. Okay, so it's wacko. And so what about Zika infection after dengue, right? Now, so far, I've been just talking about dengues and dengues, but what about Zika? Well, we can look at the effect of dengue titers on Zika, either cross-reactive titers or dengue or Zika specific titers, and it's always protective. So the really interesting thing is that now we see that antibodies from dengue are actually protective to Zika. Don't worry, I'll put this into the map. You don't have to remember everything. Okay, and what's interesting is that this actually, it aligns very nicely with previous epidemiological work that we had published, where we had seen that the risk, this was looking at prior dengue exposure and the risk of Zika infection. So there's no change, well, it actually is a risk factor, which was another paper on seroprevalence. So this is just on infection. But you actually, when you have a risk of symptomatic dengue is where you see this protection. So epidemiologically, you see protection, and immunologically, you also see protection. Okay, so that makes sense. So now what do we know? I told you already about Zika with this increased risk on dengue 2. But now I can tell you that dengue in general, has a reduced risk of dengue 1, an increased risk of dengue 2, increased and reduced risk of dengue 3, and this reduced risk on Zika. Okay, so now it's like getting complicated. Now, but what about the Zika and the other serotypes? Well, we couldn't look at that before because all we had was dengue 2. But we wait long enough. Now when the borders open, we got dengue 1, dengue 3, new lineages, and dengue 4 for the first time in 30 years. So now we suddenly could look at dengue 4. And so then, this is the crazy stuff. Okay, so this was done by Jose Zambrana, who's getting his PhD, and Miguel Allen, who's now he did a master's at Berkeley, and now getting his PhD at Michigan under Aubrey Gordon and myself. And so then so now this is the infection history. I'm gonna walk you through it. Don't panic. Okay, so we have the naive, and then we have what we call dengue immune. So these are children who come into the cohort, are enrolled with some kind of titer. So they're dengue immune, but we don't know their history, and we kind of leave them alone because right now we're really trying to get at like the specifics of it, but just, we have to have a category in them. So we call them dengue immune because they come in with anti-dengue titers. Then we have people who had 1 infection, right? So these are captured within the cohort. We have kids who had 1 Zika infection we capture in the cohort. In their past, we have them have 2 dengues because we have 1 and then another, so we can count the number. We have ones that have had dengue and Zika, right? Now we have ones that have had Zika and then dengue. Then we have those that have 2+, like more than 2 dengues, meaning like they came in immune, but then they had 1 or 2 more. And then there's some that have 2+ dengue and Zika, okay? So I'm gonna walk you through this. This is a Department of Epidemiology, right? Okay. So the first thing we're gonna do is the effect of infection history on dengue outcome. And we're gonna look at this as a dengue case, as a severe dengue, just dengue with warning signs and severe dengue, and then as a serotype specific dengue case, okay? So get used to this. You're gonna see a lot of this, okay? So what's number one? Overall risk, the dengue, so your human history is over here. This is a dengue case, a dengue 1, 2, 3, or 4 case, or the more severe category over here, okay? And so I'm gonna walk you through, so having a dengue case, again, for dengue 1, it's neutral. But for dengue 2, it's a risk factor like we saw. Okay, this is a complete cohort analysis. We also did this just in 2022, and this paper is now in press at Science Translational Medicine. And so there's, in this case, remember when it's just the symptomatic case, there's nothing with dengue 3, but dengue 4 is actually a risk factor. And it's a risk factor for more severe disease. So that's just having one dengue. When you have one Zika, and if you forget, this is like the cheat sheet over here, okay? So Zika is actually more a risk factor than dengue. It's even more a risk factor. It's higher. In fact, it makes it more, it's significant where dengue case is not. Again, but not for dengue 1, but yes, for dengue 2, and now for dengue 3, and also for dengue 4, and also for severe disease. Okay, so having one Zika is not good, which is why having one Zika vaccination may not be good. And so that's just, and so now, the dengue, remember for dengue 2, you can remember that I showed you the 2 dengue was actually not a risk factor. But strangely, it's kind of showing up as a potential risk factor for others, and so that suddenly means maybe 2 is not enough. Like people thought having 2 dengue infections was enough, and we thought that before, but that was only on dengue 2. So now we actually have new data that we're still kind of interpreting. But you can see that that's kind of what happened with 2 dengues. Now this part's really interesting. Dengue followed by Zika is a risk factor for a case, for a dengue 2 case, and for a dengue 4 case, and for a dengue more severe disease, the red, okay? Zika followed by dengue, however, the opposite order, but the same viruses, is now no longer a risk, okay? So the order matters. And this, I have at the end if you want to see it, but I can tell you that this matches the neutralization titers. When you've had dengue followed by Zika, you have lower antibody neutralizing titers than when you've had Zika followed by dengue. And we have some incredible B cell work also that we've done with CySeq and single cell sequencing on the B cells to underlie this stuff. But right now I'm just showing you epidemiology. And so this is really interesting because it shows that the order matters also, okay, because it wasn't bad enough. And so then when you get to 2+ dengue Zika, here, you know, it's essentially either protective or neutral, which is like, thank goodness, something works, right? And so now we look at the effect of the ELISA, these are the initial ELISA, the B2. So these are again that kind of the ADE curve, okay? And this is really interesting because when you look at just the dengue case, you do see this curve. And so now it's like going like this. Remember, I was showing it to you this way. Well, somehow the grad students are working out this way. You made it like this, so now we have to all go like this. So this paper goes this way. So here, this is now the curve, and you can see it also over here with, you know, the up again at 21 to 80. So that's still true. But now look at this really interesting thing. When you have dengue 1 or dengue 3 again, remember early on you can get this ADE, but really it's mostly protective. Okay, so you actually have it's not really a curve, it's more like a line. But then when you have dengue 2 or 4, you see that curve. Okay, so it's really different by serotype. Okay, and this is again the incoming serotype. No way. Okay, sorry. Go. Okay, that's clear. Okay, so I asked for the 10-minute mark. So here we go. I'm just taking my time to make sure you understand. Okay, so I made the event complete, so now we just put the whole right in the bed. Right? Are you with me so far? Okay, good. So now we squish everything together in flaviviruses because we're just going to see, you know, when you put everything together. And here, whoops, what we see is when you're flavium, meaning dengue or Zika, you get more power that way, right? There you can see again that it's a risk factor, but not only for this, you know, 2009, but also for honest to goodness DHF DSS, because now you push that together, you have enough to have these rare outcomes, which are the severest disease. And then again, you see that decreases the risk of dengue 1 and increases the risk of dengue 2 and 4 and neutral for dengue 3. So that's kind of like the final thing. So then we get this. Okay, so but now you totally, this is not scary to you because you fully understand everything I just told you, right? Because I explained one by one by one, so now we can actually put this all together in this really cool thing and blow our minds. So this is like, the FDA does not like this, okay, because this is complicated, and this is not just like, oh, vaccine is not a problem, vaccine, we need one, but we have to be careful, okay? So what are the implications? I just mentioned vaccines. Well, it turns out that the dengue vaccine trial, we knew this coming, they went ahead and did it anyways, they put it to the Philippines, even though they could show that when you are seropositive, meaning when you are an individual who already has some kind of dengue immunity, you end up being protected because it gives you more immunity. But their particular vaccine, which was a live attenuated vaccine to all 4 serotypes, had unbalanced immunity that they couldn't see with the standard new test. Because there's all this stuff underneath, the quantity and the quality, all this stuff. And what happens is that it actually, this is bad, this is where your placebo does better. So the vaccine is actually a risk factor for hospitalization. And we saw this coming, we told them, they saw it themselves, and they still put it into the Philippines, 800,000 children. And there was issues, and they're on trial for homicide, okay, it's a no beach in the Philippines, no one can go back there now, so it's like a real problem. And it really set back vaccines because people were scared, so then Takeda comes along, and they do it, and they don't live attenuate, and they said, we would never do anything like Sanofi, theirs is also unbalanced, and so the problem with Sanofi is it didn't protect against dengue 2, it was a dengue 4 vaccine, the Takeda is essentially a dengue 2 vaccine, and it doesn't protect, it looks like, against dengue 3, and they've still gone ahead and registered it, and it's still being put into countries. Okay, problem. So implications. So dengue vaccines, differences in efficacy in phase 3 trial may be explained by lack of protection and enhancement, right? So remember how I showed you there was no effect because when you have all the serotypes together or 2 different things going on, it looks like nothing, but there's really 2 different phenomena underneath that, okay? Same thing by serotype. You need to look at the serotypes separately because some are up and some are down. And if you just look at them together, you get a straight line. But it's essentially misleading because there's stuff underneath there. For Zika, if you have a monovalent Zika vaccine that does, what a natural Zika infection does, you can actually increase risk of subsequent symptomatic and severe disease. There are ways that we can talk about the question and answer to get around this, but there are next generation immunogens, which are really exciting. They're void generating, potentially enhancing antibodies. And then, even Zika protection can matter across different constructs. So it's really tricky. Okay, so that's kind of what I wanted to tell you. And I have this little coda in the last minute. So what I told you so far is this super complexity, but then we can really with these cohort studies, can really understand what's going on year by year. And what I've shown you so far is the kind of imprinting with a dengue or a Zika, any dengue or a Zika infection, and then differences by the incoming serotype, right? Now the last frontier is really the landscape of inapparent infections going backwards, right? So what's cool now is that we can now, with this new method we have, this is by Sandra and the postdoctoral lab and Fernando and Jose Victor, so they've set up this really nice Luminex system where we can look, this is not neutralizing antibodies now, we're combining antibodies at the level of the serotype specific, but then what essentially I'm going to focus on is looking at antigen specific antibodies by serotype. And then the next level up are epitopes. Okay, so this is how we can look more and more and more higher resolution. And we have ways to do all of this, but I'm going to focus just on one. And so the problem is that if you want to look at all these different epitopes and we want to look at different, even just different antigens, so we want to look at the E antigen, which is the envelope, and then there's this E domain 3, which really allows you to serotype because it's where most of the serotype specific antibodies bind. And then NS1 also can help with deconvoluting serotype. But this would be 15 wells by every subject, by every time point, by every dilution, and it just becomes completely impossible. But if you push them all together, we can do this 15 antigen in a well with this Luminex-based system, which allows us to do 1 well for all 15 antigens with 1 microliter of serum, so we can suddenly make that work. And when you do that, what we did is we took symptomatic infections, so 64 symptomatic infections of the different serotypes. And then we ran this Luminex, we knew what they were, which was the serotype over here by PCR, and which is over here. And then we just do unsupervised clustering by what we call the serotype should be by this Luminex method. And then we just let it go. And you can see that it's 100%. Essentially, we can do this really nicely to serotype by Luminex instead of by PCR. Okay, and this is, PCR is looking at the molecular RNA, and the serotype is just looking at the imprint at the antibody level. But this is really exciting because you can just use that now to serotype. But that's, you know, who cares? These are symptomatic infections, right? That we know that because we have the PCR, we don't need this. But where we do need it are for inherent infections, right? Where we've never been able to really, I mean, you can do this by nu, but it's impossible to do that many infections by nu to all four by every dilution. It's just, you don't do it. And so now we did another group, 86, and these are inapparent infections that we did do the neutralizing antibody, and these are primary, so it's easier, first step. And then we also, so this is what the serotype by neutralization across here, and then we do it by the Luminex, and we can, it works really well. So we actually by have 100% sensitivity, specificity, and diagnostic accuracy when we have, you know, something that's readable against, you know, the gold standard. So what did this allow us to do? So we took 40, this is the first stage, was 46% of all of our inapparent infections backwards, okay? And so what we're seeing here is this is, well, it'll come up, sorry, right here. And you can see what, you know, the symptomatic, we had all dengue 1, this is all dengue 2. Remember how I showed you that nice little piece, they're all pretty, one big, and then these are the numbers, so you can see that like we have more confidence here because this is how many we serotype, and we have fewer here, right? So they're kind of grayed out just because we're not, you know, we don't have to grade. So here we can do it by year, and this is so interesting. Look at this. The symptomatic was all dengue 1, and look what was going on underneath the surface. So that's what was actually circulating. So I've been like bullshitting people all these years, saying like, look how pretty it is. Like, what happened to your plate? And God, this is. But so this is a statistical method. And this is actually also imputed. So this is like chapter 2 of Victor's thesis. So we had to do all this fancy imputing and blah, blah, blah. So we have reality, and then there's reality plus imputation. Just the way the non-modeler looks at it. Okay, and this is statistically different. And you can see here, again, we think it's all dengue 2, and in fact, all this is going on. We think this is all dengue 3 almost, and it's actually not. Okay, so it's a lot more complicated. And this is showing here where I just told you before, look, here's my, so the gray is like all of the infections that in the cases that we've had, and then here is by serotype. So you can see that the peaks are really, you know, what is one serotype. But here you can see all the infections. And in dengue 2, it really was only dengue 2 in 2019. But here you can see that there's actually all this other stuff going on. So it's not as clean as the symptomatic infections, which is really interesting. But this tool now allows us to essentially go back and also look in a primary infection. So that's the tool that allows us. Now I'm going to do the very last coda, which is saying what is the clinical you know, what matters about these serotypes, and the serotypes are really different clinically. So we can look at here for the first time is the ratio of symptomatic outcome by serotype, and here we're looking at the inapparent versus symptomatic, and no one's ever done this because you have to know what the inapparents are by serotype, right? Which we can now do. And so what you can see is that compared to dengue 1, dengue 3 is more virulent, right? Because these are primary infections. Usually you don't talk about the word virulence with dengue because it's partly your immune system and so it's not really the virus only, but these are primary infections. So all is going on is one virus in your body with no antibody. So we can really say that there's something about dengue 3 which is more virulent in that it is, you know, to the, you know, is more likely to odds ratio 2 here. It's called a pool odds ratio. But then also if you look at dengue severity, you can also see that dengue 3 in primary infections has more severity than the others. And this is and you can see that this, you can really see when you look by serotype, this is the inapparence and then the, you know, the kind of dengue without warning signs, with warning signs, and severe dengue. You can see that dengue 3 is a baddie. And what Takeda has been running around saying that dengue 3 is not a problem. And they've been literally going country by country. I know because they go to Nicaragua and since I talk to the minister all the time, she's like, you know what they just told me? And I go, you know, so and they go and they say, oh, dengue, Sanofi was like dengue 2 is not a problem because, and now Takeda is running around saying dengue 3 is not a problem because it doesn't work in their vaccine, but it is a problem. And so what we've been able to do is also with another study, which was looking at our hospital study and our cohort study, and the ones from the cohort go to the hospital, 2,400 dengue positive out of 14,000 people. This is like really, really complete. And I just want you to see, this is different. Like this is, these are all severes by 1997 or by 2009. And you can clearly see that of the severe disease, dengue 1 and 3 have severe disease and primary infections, and you just don't see that with dengue 2 and 4. They're different viruses, okay? They're different viruses, and they can cause different types of disease. And that just shows you. And then the last slide is that if you look at proportion of severity compared to dengue 1, if you look at the 1997, it's dengue 2 and 3, and if you look at 2009, it's dengue 3. Dengue 3 is not a good one, okay? And we can talk about why the clinical different, the definitions are different, but to take home is the following. There's a complex immunological and neurological interactions among viruses. Are you convinced? Okay. Dengue serotypes are different viruses. It's not 4 things are the same. They're actually different with distinct clinical and immunological profiles. You need to take flavivirus serotype and order even of infection into account in repeat infections due to this imprinting phenomenon. Immune correlates, I didn't show you this, but we have a whole thing on immune correlates, like biomarkers of protection and risk that actually also unfortunately vary by serotype. It's really interesting. Dengue 2 cares more about neutralization, and dengue 3 cares more about virolysis. I mean, there's actually really interesting, but there are differences. And there's important implications for vaccine design and evaluation. An unbalanced live attenuated vaccine can increase the risk in dengue naive. So you really need to consider these next generation mutagens. Companion diagnostics are important to avoid vaccinating dengue naive individuals. And dengue or Zika vaccines may not increase the risk in individuals with complex infection history. So like one piece of good news, I'm telling you. And now to date, we really talked about imprinting by infection history and the role of the incoming serotype. But the next frontier is imprinting by dengue serotypes and order of infection, which is what our next PO1 is. And I hope, Mark, you're on the review committee. No, I'm just kidding. No, anyway, I wanted to thank my incredible group. So Sandra and Navita and another one, Elias, and these people just worked on this project. A lot of the new correlate work is done by others. The pathogenesis work. Leah Katzelnik, again, she's now our own group, but we keep having wonderful collaborations. Some of our 83 was sourced from Vaccinology from Kumar. And this is the folks in Nicaragua. There's actually 225 people who work on our studies in Nicaragua, now joint with Aubrey Gordon, who runs our flu and COVID and respiratory work, and she's a former student now, amazing professor. And so this is just some of them, but led by Dr. Anibal Maceda at the National Virology Laboratory and SSI, which is based there. Anyway, so this is our folks at the health center and at the hospital. And the kids, these kids actually are now pretty much grandparents because I took this picture in 2004. Anyways, it's really hilarious. I saw the picture, so anyway. So thanks to our kids and thanks to you for showing up and give them time.
I think we have time for this couple.
Stunned silence. This is like after the Zika epidemic. Okay, yeah.
Thanks, that was awesome. I was wondering, if you could say anything about the time between the infections.
Yes. I can, except there was so
It makes it more complicated.
So yeah, it is, although it's really interesting, but I don't really understand why. So we did studies and other people have done this as well. And what we see, well, Saban in 1952 did these very classic studies where he took military recruits and literally just injected them with dengue to see how long they would be protected. So that's kind of where we got this idea that 6 months is kind of like one area where you're protected in a homotypic infection, but not for heterotypic. But in epidemiology, our group and a tri group were pretty much at the same time found that you were protected for about 1.5 years to 2 years from an interparent infection, and then about 2 years and some from a symptomatic infection, and then a little bit later for a more severe. And so that bolstered the idea that that you have waning antibodies, and that when you get into that 21 to 80 kind of danger zone is where the problem is. It got more complex. We have a couple of papers on antibody dynamics, the one in Central Medicine 21 and another one where we looked at it 3 years later. And like after primary infection, they're completely solid. Like it's so strange that we expected it to see it go down, but in fact, so everything we do blows up dogma. It's crazy because it's just like the data is the data, right? But like what we thought is that you have it going down after a primary infection and stable after a second infection, and instead what we see is really rock solid antibodies after the first infection and then some waning after a second infection, probably because it's so high, and so we have various, and so us and several other groups have also seen the same thing, and we think that there also might be kind of a geographical focus in the sense that you kind of see every kind of 2 years or so, you just have new serotypes coming in. So it could be that what we attributed before to an individual level risk is more the population level risk of having new serotypes come in approximately every 2 or 3 years. You see what I'm saying? But we are also, so this was the inhibitional lysa titers, which are really solid, and they were mostly to fusion lupin PR antibodies, but then we've also done it with Luminex, which are to the others, and so to anything envelope-ish, they're very solid titers, which makes sense because it's known, like Mark Slipka and others have shown, that B cell immunity is really strong when you have multimeric antigens. But the NS1, which is by itself, I mean, it's a hexamer, but it's pretty much by itself, that's when you start seeing some reducing. And so now maybe the NS1 becomes important in all this stuff, so we're just kind of unearthing this as we go along. And now we have more and more, we've made more tools to look at specific epitopes. And so we're also looking at neutralizing. So we're kind of unpacking this whole thing, because we're still trying to figure out what is it that is, why is it that you get that kind of protection over a certain amount of time that goes away when it's not like obviously all your antibodies going away, because they're actually very, stable. And so there's a lot kind of going on to figure out that. That was kind of a long answer.
How does your multiplex assay perform for non-naive children?
Right. So that's the next thing. So you can do it in a second. By the time you're in third infection, forget it. It's a mess, I mean, but and some that are second also. So that's what we're trying to do right now. And so we, I'd be happy if we could like figure out 60% or something, which is, but so yeah, so we're doing that very carefully. We have people that we know with infection is, so we're like training the system on them, and then we're looking. And we're trying to get something where we can get some amount, because even by newts, it's hard to tell. Some places are clear, you have 2 things that are up, and others where it's like a mess. And so by the time you're at third, unless you know the infection history, it's very difficult to kind of pull that apart. But it gets us a long way kind of down the runway, yeah.
How did you choose the targets for the Luminex assay?
How do what?
How did you choose the targets for each Luminex assay?
Yeah, so it's been, I mean, there's only like 3 structural proteins, right? I mean, so it's not like the super complex bacterial parasitic genome, right? For like malaria, epidemiology is super complex. But essentially the E domain 3, it has the most type specific epitopes. And so there's another way that Ralph Berkeley we collaborate with has made these epitope swap viruses, so we can actually look at neutralizing antibodies to a particular epitope. There's like a dengue 2 neutrotype specific epitope, like in a dengue 4 virus. And we've done a couple of papers looking at that in our cohort. But for binding, really ED3 is the best. And then NS1 can kind of help. And so actually, the paper just came out today, and it was accepted in Mexico. Will help look at that in pregnant women for Zika and dengue to differentiate, and the NS1 is helpful. So kind of those 2 are good antigens. Yeah.
In Africa, we're dealing with both the Zika and dengue, but also yellow fever, both vaccine and natural infection. Do you think yellow fever plays a role in imprinting?
Yeah, no, actually, it looks like there might be protection by the yellow fever vaccine more than anything. But it's interesting that you mentioned this. So, so we have a grant that just ridiculous, a 10th percentile grant that I had to put in again because the chain line is 8%. But okay, we resubmitted a grant on this very interesting story of can you use NHPs in non-human primates in relation to humans for the next emerging flavivirus. Okay, and so the people who are collaborating with Tom Friedrich and Matt Aliota have shown a really nice NHP model for dengue and Zika, but they also went to sponweighty, which is kind of hasn't hit the human population very much except in Africa. And they showed in their NHP model that Zika protects against sponweighty, but sponweighty does not protect against Zika. So they've got another asymmetry going on. And so what we did is that we said, can we use the non-human primate to really predict what's going to go on in humans? And so far, NHPs have kind of anecdotally been quite good, very good with humans, but it's like this paper, you say so, I say so. So it's not really like solid. And so this grant is really cool because we aligned, so they already have a bunch of NHPs like Zika, dengue 3, they do the Zika respond waving, so they have all this, of course, they can challenge respond waving, but we can't. But they can get, so we have all this stuff that we can actually match in, so we've matched our human cohort with the NHP. And so we can look at every, first infection, second infection, and then the immune response is von waning. And then we can match, we can see what the clinical outcome is in the NHPs, and we can see if it matches what we see in humans until that point. Because the ideal thing would be to have like, and these are not lethal in NHP studies, right? They're immunological studies, and because the monkeys don't get that sick, they have like a viremia, and then they have, but the idea would be, could you, if you had a new flavor coming in, could you kind of try it in the NHP model and see whether you can expect there to be an enhancing or a protection. And so we have all the statistics to really look and see, like, are the immune responses similar in NHPs and humans, because that would be great. And not just kind of like, okay, I'm gonna do it on like one dengue, like in the complexity of multiple flavies circulating, can we still see this effect against a new emerging virus? And so that's just speaking about, I don't think that yellow fever is a thing, but others definitely could be a problem. And that's what we're kind of trying to get ahead of the ball on that.
Time for at least one more.
Why do people go for dengue serotypes?
Yeah, and so they have jumps multiple times, and not even that long. It's like these are all coming like 200 to 500 years or something. It's not super ancient, And they jump Sylvatic. There's a bunch of Sylvatic, and they come in and then they establish. And I think that there is this thing where it's the partial enhancement and protection, and so they have to balance. They kind of want to be close enough so you can get a little bit of enhancement, but then not too close because then you can get neutralization. And so it's kind of like, it seems like 4 is the magic number, because there was one claim of a dengue 5, but it was really very close to dengue 4, so it probably is still within that serogroup. Anyway, so it seems that it's kind of the virus finding space in, kind of genomic, immunological space, whatever you want to call it, where it can land, and then kind of hold on in relation to the immunology, kind of the serological responses of the other serotypes. And so it kind of balances out with the 4. Yeah, good question.
Okay. Well, barring anyone, thanks.